Hi, my name is Dima, and in today's meeting exam video, I'll be reviewing the features of Watson and Crick's DNA structure. Before we begin, let's talk about nucleotides, which are known as the building blocks of DNA. Nucleotides are made up of deoxyribose sugars, a nitrogen base carrying the genetic material, and a phosphate group. If we look at this 5-carbon sugar, we can see there's numbers 1 through 5, and these represent the numbers of ribose carbons. The 1' is attached to the nitrogenous base. The 2' distinguishes DNA, which has an H group, from RNA, which has an OH group. The 5' carbon is attached to the phosphate group, and the 3' carbon bonds to the phosphate group on the next 5' phosphate group. Phosphate groups create the sugar phosphate backbone held by phosphodiester bonds. These groups give the DNA a negative charge, which we'll be talking about shortly. There are 4 types of nitrogenous bases. There's adenine, guanine, cytosine, and thymine. Adenines and guanines are purines, having double ring structures, while thymines and cytosines are pyrimidines, having single ring structures. Adenine binds to thymine, while cytosine binds to guanine. A good way to remember this is that A and T are pointy letters, and C and G are curvy or circular letters. Adenine binds to thymine using 2 hydrogen bonds, while guanine binds to cytosine using 3 hydrogen bonds. Hydrogen bonds hold these nitrogenous bases together, they're weak and aren't the main force joining the 2 strands of DNA. The main force which holds and stabilizes these bases together to form the double helix are hydrophobic interactions, which are maximized due to the base stacking. Hydrogen bonding provides specificity. DNA structures have 2 polymeric strands which are antiparallel, meaning each strand has a 3' end and a 5' end. The 3' end of one nucleotide bonds to the 5' end of the next. One strand goes from 3' to 5' direction, while the other goes from 5' to 3' direction, making it antiparallel. Nucleotides are directionally linked. The DNA can be right-handed or left-handed depending on the DNA strand's helix direction. If the helix is pointing up to the left, it means that it's left-handed, and if the helix is pointing up to the right, it means that it's right-handed. Almost all DNA is right-handed, but it's possible for it to be left-handed. Let's talk about the grooves which we'll see on the helix. We have minor and major grooves. The grooves within the helix alternate between small and large. The small grooves are known as minor grooves, while the larger grooves are known as the major grooves. The distance present in the major grooves allow for the proteins to interact with the nitrogenous base pairs. And lastly, let's talk about the DNA's melting point. Let's go back to the negatively charged phosphate group, which I mentioned earlier. The phosphate backbone's negatively charged state acts as a destabilizing force within the molecule. If the DNA is added to a salt solution buffer, the negative charge would get a little neutralized, which reduces the destabilizing forces, making the DNA held together more tightly, and the amount of heat that would be required for it to break apart the molecule would increase. The midpoint of thermal denaturization is called Tm. This is basically just the point that it takes 50% of the DNA to be denatured or broken apart, and using a salt solution buffer increases the DNA's Tm. Here are references for this video. Thank you for watching.